Dear students, now we are going to see about different types of media and culture techniques that are available in the microbiology laboratory. Before that, let us see about the introduction about the culture media. A culture media is nothing but it giving a nutrition that is for the preparation used for the growth, storage, transport and identification of microorganisms in the laboratory testing. So it provides all the necessary nutrients that are necessary for the growth of microorganisms. That is nothing but the culture media. Basic composition of media, it contains water, which is the essential vehicle for the nutrients, and peptone, which is the digested protein, and it is the source that provides nitrogen and amino acids, and carbohydrates, that is the energy source, particularly glucose and lactose, and minerals, for example, sodium, potassium, magnesium. So these are all the necessary required basic nutrients that should be available in the culture media. And there is another one important nutrient composition that is present in the culture media is the agar, and it doesn't provide any nutritional value to the media, but it will act as a solidifying agent. The agar is nothing but the polysaccharide that is obtained from the red seaweed Gelidium. That is the family of that red seaweed Gelidium. And the key properties of this agar is it may solidify at 42 degrees Celsius, which is perfect for pouring, and it melts at 95 degrees Celsius, and most bacteria cannot use the agar because before that agar some other components are used which are readily used by the bacteria. So after that, the agar is the best source for the solidifying agent, which keeps the surface solid as much as that. And we can classify the bacteria based on the consistency, and we can classify the bacteria if the media is in solid condition by adding agar means it is called solid media. If we use media without the agar means it is called as a broth, and we can say it as a liquid media which is no agar. And based on the composition, nutrient composition, we can divide this media into two types: synthetic media and complex media. The synthetic media, it uses exact chemical formula which is already known compounds, and in complex media which contains the extract, that is beef extract and yeast extract media. And based on the application and functions, we can classify the media into three types. One is basal and enriched media, which is used for the general growth of the microorganisms, and selective and differential media used for the identification of the microorganisms, and transport media is used to transfer the media. So based on the consistency, already we have seen the example for solid media is the nutrient agar, and the liquid media we can say it is a nutrient broth because we didn't add any agar as a solidifying agent. And basal media, the example for basal media is nutrient broth and nutrient agar, and it supports the growth for the non-fastidious organism because it contains basic simple nutrients. And the example for enriched media are blood agar, serum agar, chocolate agar. We can add some enriched components like blood, serum or egg, which is used for the fastidious organisms growth, and bacteria with complex nutritional needs which only grows in this enriched media. So example for that enriched media is blood agar and chocolate agar. And then the selective media, for example for selective media is the mannitol salt agar, that is it contains high salt which inhibits the growth of non-staphylococcus bacteria. It contains inhibitory substances, either it may be an antibiotic, dyes, salts, which suppress the unwanted microorganisms and encourage the wanted microorganisms, that is the desired microorganisms. In differential media, which allows multiple types to grow, but it distinguishes between what we need, that is we can differentiate either McConkey agar, lactose fermenting bacteria, and non-lactose fermenting bacteria, for example McConkey agar. And then this is an example blood agar, which we can differentiate the beta lactam and alpha lactam and gamma lactam bacteria. Example for this blood agar is Bacillus subtilis. Staphylococcus identification can also be can be done by this, and mannitol salt agar that used to isolate and identify the Staphylococcus species. And by using this, Staphylococcus pathogens and Staphylococcus epidermis, which is non-pathogen, can be isolated. And specialized media types, there are also some media which can be used for the to transport purposes, to keep the bacteria which are alive during transport and without damaging or without destroying them. So for transport media, several medias are available. Starch transport medium is the best media that is used for transportation purposes. And there is another media, Robertson's cooked meat media, which is used for the especially for the anaerobic microorganisms. And these are all comes under the specialized media types. Thank you, students.